What I'm going to talk about is SOPs, or standard procedures for multimodal informatics. So helping us, things we can do to get to insights and innovative therapies faster. I was thinking about maybe, instead of saying beyond the standard of care, because that's kind of ambiguous and open to interpretation. Based on Dr. Castro's comments, maybe it should be beyond evidence-based care and going to science-based care. I've been doing bioinformatics in various forms for over the past 20 years at University of Chicago, some various biotech companies, now at Illumina. And like Pete mentioned, I've been involved in 3 of these RTTP cases so far. And there's some recurring themes that from over the past 20 years, and then also during that I've encountered during these RTTP cases as well. And it's really a lot of it comes down to plumbing and organization, so we can make use of the data that's already available to us and the tools that we have, so we can get the most value out of this data. Some of these recurring things are standardization is hard, but it's essential. Orthogonal data sets are key to understanding biology. You need the RNA, you need the DNA, you need the longitudinal context is so important, as you saw with what Marvin was presenting there. When we have a tendency to just look at a single snapshot and have a single assay or a single test and try to figure out what's going on, but you need that context to get the signal out of the noisiness of biology. And so even though this organization, takes work and it's boring and it's kind of tedious, it's really what enables us to come up with the creative solutions and be collaborative. When you've got all this data, this wealth of data, you have to be able to organize it and understand it and have good data provenance, know where that data came from, and have all that metadata as to where that data came from. So what I'm going to talk about today, here's my agenda. So first, I'll give a definition for what I mean by an SOP for informatics. It's basically standardizing the methodology for gathering data, both the primary and the meta data, the analysis, the secondary and tertiary. So in secondary analysis, and we're talking about aligning, variant calling, generating RNA expression levels, and then the interpretation of it. And so, like Pete mentioned, I was there at the first hackathon. And what I've realized is, there's definitely, we don't want to lose that hackathon, mentality to these cases. But to enable us to get to that, there's kind of a gap to get there. So, like Pete mentioned, we had 40 or 50 people on site. We all got together. We got the background of the case. And then we all kind of broke it up into groups and started trying to figure out what to do. And I think there are certain things we can introduce, these SOPs, so that we get to a place where we can start prioritizing and evaluating options faster. And so basically where I think I propose that we start off is really with, where the standard of care and the latest approved therapies leave off. And really, I was at ASCO last weekend, and there, one of the things that Illumina was advocating for and providing education about, as well as a lot of the other folks at the conference, was comprehensive genomic profiling. And that's basically what the Personalis Next ID, provides, I think, largely. But there's many other, comprehensive genomic profiling approaches out there. And it's becoming part of the standard of care as well. But so how we get from, where the latest therapies, that Oliver was talking about, to the ability to do really creative, innovative things, I think we can get there faster and more efficiently by introducing some of these SOPs for informatics. And I'll provide a little bit of some details from Shirley and Vanessa's cases to kind of before getting into talking about the prerequisites for these SOPs. Basically, this is if I often I think of myself as like a data and bioinformatic a plumber of data. And if you want to have good plumbing, there's some certain concepts and certain ideas that I think we should embrace. And that we are in the process of embracing. Then I'll talk about some of the SOPs that are actively being developed, but in truth, these are really more SOP components and the bioinformatics pipelines that can be stitched together to create a larger workflow. And then I'll talk about some of the planned SOPs, and then I'll just follow up by why this is important and why this matters. So this is kind of this is a subset of the samples that Marvin was presenting to you. This is 32 of the samples. It's a mix of Shirley's samples and Vanessa samples and some control samples. And it's just a hierarchical clustering plot from Python, SciPy. And what you'll see is this is from like one snapshot in time. You see you got one of Shirley's samples up here, and then you've got another of Shirley's samples down here, and then another one in the middle, and you've got Vanessa's kind of all over the place. And then you've got some control samples next to ovarian cancer samples. You get some little patches of clustering. And the reason is, in this case, we're looking at all those 22,000 transcripts at that single time point. If we were to look at certain pathways, we would see better clustering. If we were to remove some of the batch effects, due to whether it's fresh frozen or FFPE, if these were done over multiple years, they're done on different sequencing platforms. They have different amounts of tumor cells within these samples. So these are all things that, without that additional metadata, without that longitudinal, it's very hard to separate. And so I think in the first hackathon, we kind of jumped in, we got this data, we got tools, it's very hard to come up with a suggestion of a therapy when you're looking at it. And so we need something to bridge it to before we get to that hackathon mentality. So going to the other end, further upstream. So here at ASCO, our medical affairs team at Illumina put together this very useful educational asset. And feel free to check it out. It's, the illumina.brightact.com. And you can go and you can click on any one of these types of cancer, and it will bring you to a table that shows here are the various biomarkers and variants that have approved therapies or suggested therapies based on those biomarkers. And it's really interesting to look at, and you can click on it further, and it'll provide some evidence to show, the responsiveness of these. And if you run one of these comprehensive genomic panels that can indicate, what markers are present, you typically in the clinic, report will be generated. It looks something like this, where you get a TMB biomarker, an MSI marker, a GIS score, which basically is an indication of whether a tumor is HRD positive or negative. And then you'll get an indication about markers in specific mutations or variants in specific genes in that panel. And then from there, a variant scientist will go through and decide which information, from this report gets in the report that's given to the physician so they can make decisions about treatment. And this is kind of the standard of care, the latest today, where you take this test, but there's a whole lot of data underlying these comprehensive genomic tests that is not available to the clinician. And but in these really challenging cases like Vanessa's and Shirley's, where ultimately it's going to be essential to have that data and have that metadata about it to be able to come up with additional suggestions of treatment. And that's really what Fareshta and we've been working on. We've been taking the underlying data. We've been kind of going beyond what you get and from the high level report and kind of digging into the weeds and the details. And so we've loaded up, the RNA-seq data into the Illumina Connected Analytics platform, which is a cloud-based platform for doing secondary analysis and running some Dragon-based pipelines. Those in the bioinformatics community may be familiar with it. And when we ran these RNA-seq analysis, here's like an example just of some the highly expressed genes in Shirley's case compared to some of the control samples we had. And so now there's one of the things you can do kind of beyond the normal, standard of care is you can take this and you can compare that signature that we got and you compare it to a much larger knowledge base. And in this case, Illumina has a knowledge base called Correlation Engine, which has like 25,000 studies in it where they've been curated, and the data analysis for various types of analytes, RNA, proteomics, DNA, has been normalized into all these studies have basically either treated versus untreated results or disease versus healthy results, and normalizes them so you can take your signature from your data set and then compare it and see what correlates with it, thus the name Correlation Engine, or what's anti-correlated with it. And you can do it in different contexts. So you can do it in the context of different drug interactions. You can do it in different types of tissue. You can do it in types of different pathway. And basically what it's doing is it's very similar to a gene set enrichment analysis. It's using a running Fisher algorithm to basically say which is how of these various in all these 25,000 different studies, which one has a signature that correlates with your data or is anti-correlated. And when you're looking for maybe off-label drug use, one of the interesting things to do is to look at compounds that have a anti-correlated signature with yours. And when I did that with Shirley's data, the top negatively correlated signature was associated with this particular drug here, which I'm not going to attempt to pronounce. But it is a mTOR inhibitor. And I believe I think I saw on one of Fareshta's slides that there was an mTOR inhibitor that was suggested as a possible treatment. So I was happy to see that. So this is just the type of thing we can do to get to possible, treatments by taking the underlying data and running some, very simple statistics and uploading it to existing knowledge bases. There's such a wealth of data out there. So, if we're not with evidence-based medicine, but, we can definitely go to data-based and science-based medicine fairly if we are smart about how we organize our data and we streamline the analysis. So one of the other things that came out of the RNA-seq analysis that we ran was both in Shirley and Vanessa, there was this is from the last time point we had for each one of them. A large number of fusion transcripts came out of it. Now this also could be possible to some of these fusions undoubtedly are due to the treatments they underwent, chromosomal rearrangements and things from the treatments. But fusions are a ripe area for different targeted therapies. Now, this paper here shows in high-grade serous ovarian carcinoma that this is a very targetable fusion. Now, Shirley did not have this specific fusion, but in the there was evidence in her RNA-seq fusions that she had several that involved this AKT2 signaling kinase. So this is another place where I think we can go and look for leveraging existing therapies or off-label drug use to target these fusions. If you go and look at, like, for example, in the My Cancer Genome knowledge base, you'll see that there's 941 different fusion biomarkers that are associated with either available drugs or clinical trials. So fusions, I think, are another place where we can turn to look for innovative treatments. So I'm going to change gears here a little bit. So we have all these tools available to us. We have all this data available to us in these other knowledge bases, and we have the ability to generate additional data. But going back to my original point, how we organize this data so that we can get to new innovative treatments is really key. And so so in terms of some principles in terms of data, we should in setting up our environments, the data, we should have it so it's entered once or 0 times. 0 times if it can be derived elsewhere from existing data. And one of the reasons I say that is because what we still see often, probably one of the biggest challenges that that people I work with is with all the amazing technology we have, still one of the biggest challenges is just getting data from point A to point B, or from program A to program B. And because every time you have to move it or enter it or re-enter it again, which happens oftentimes in our workflows, either data, it introduces errors into the metadata, or metadata gets left behind, and you end up not really necessarily being able to know what the source, that you don't have a single source of truth. So, if we could just get to the point where we're entering the data as few times as possible, that's and moving it as little as possible. There's a set of principles, FAIR, that your data should be findable, accessible, interoperable, and reusable. And so when we're setting up our environment for RTT projects, and there's a lot of different ways we can do that, we should definitely keep in mind these FAIR principles. And we should definitely always, look to leverage existing data as much as we can, like TCGA, other knowledge bases like My Cancer Genome. And there's increasing ability among institutions to be able to share their databases with one another. So there's a large movement where academic medical centers can opt into sharing how they've classified a particular variant and what the treatment was with one another, which I think is a wonderful thing. When it comes to the tools, we need to make sure they're modular. We make sure that they're versioned, and they need to be transparent as well. And these can be commercial tools as well as open source tools. But we need to understand, the assumptions they're making, and they need to be benchmarked or benchmarkable, so we can, as different versions come out and we want to analytically validate them. So having them benchmarked is really key. So we can measure our progress, measure changes, and know which tool is the appropriate one to use. When it comes to the environment, and I'll admit I'm very cloud biased. I'm a cloud biased person, but whatever it is, it needs to be secure. Of course, it needs to be scalable. It needs to be collaborative, and it needs to be supportive of different roles. So we have on this call here, we've got folks with all different backgrounds, and I think they're all, so whether you're a clinician or, a biologist, a programmer, you need the environment to be supportive to access that data and contribute, to this progress. So I'm going to talk about, initially I called them SOPs, but then when I thought about it, these are components of a larger workflow. In my biased, view of the world, like these are largely secondary analysis pipelines that take data from sequencing or take data from CyTOF data and transforming them into something closer to, can be used in clinical practice. But they're not the entire SOP. How you, filter any data, how you organize that metadata is part of that larger SOP and an important part of it. How you do QC on that data, how you remove batch effects. So it's much more than, the overall SOP is just much more than the secondary analysis pipeline itself. And so Fresh and we've been kind of developing these. So the RNA-seq pipeline, the single cell RNA, we've run some samples through a single RNA pipeline. We've got some whole genome germline data we've been analyzing, tumor normal, looking for the ability to screen look for viruses and the effect that viruses may be having in the evolution of a particular tumor environment. So these are ones we have in development. The ones that we have planned, or we've done a little bit of tinkering with some of these, is longer. So the ability to, take those CGB reports and analyze them. One, I think, exciting development that was, was heavily, talked about at ASCO was the ability to use liquid biopsy, for monitoring of, post-surgery, post-chemo, or post-targeted therapy, to be able to monitor a particular case. And I think ultimately and I'll talk about this more at the end, we want to be able to, have workflows that are iterative, where it's not just we suggest a therapy and we cross our fingers and hope it works, but we can, apply a therapy, monitor it, and then iterate on it. And so that's one of the reasons why the speed of streamlining our analysis is key. We want to be able to characterize the microbiome. We want to be able to do, characterize the immune system in many, many different ways. You can see my sequencing bias here, but being able to do it through other methods as well is important. We did some, and I think later today is going to talk about personalized cancer vaccines and having pipelines for neoantigen identification, personalized cancer vaccine design, looking at epigenetics, proteomics, and then, and this is something Marvin, I probably would be interested in. I could definitely learn some things from him is on the data science part of it, like, having standards in place for how we do batch effect detection and regression, feature selection, model building, evaluation, and selection. And the wonderful thing is, there's so many great data science, machine learning tools out there that are really not that hard to run or implement. But the key is you've got to have well-organized data going in to have meaningful results coming out. So I don't know, how are we doing on time here, Pete? Doing all right? Okay. So in terms of like the significance of why, it's so important to get this plumbing right is, these patients don't have a lot of time. And most of the people, that are working on this are volunteers. They don't have a lot of time either. And so we need to be efficient with how we analyze this data. We need to make sure we're not reinventing the wheel. We're leveraging the existing tools and software that's out there, and helping us get up to speed on these cases. So we can quickly see that longitudinal timeline, see what interventions, what targeted therapies may or may not have been tried, and then quickly understand, in the cases where there has been a targeted therapy or other therapy applied, we can quickly understand why it stopped working, or why it didn't work in the first place, so we can make an informed decision about moving forward. It's not just this was tried, but actually understand the mechanism of resistance. And like, so it all, yeah, it comes down to being able to, fail faster, iterate and improve, build and share our knowledge. In what keeps me from, actually ultimately taking that job at the garden center at Home Depot is the idea that, we're going to be able to, within, a week or two of time, have a new patient come into the RTT program, have all their prior data ingested, run it through this standard set of pipelines, and be able to suggest, like, here's a prioritized list of, drugs to try, or known ones that are approved, ones that are off label, maybe design a personalized cancer vaccine, monitor that treatment, and then be able to iterate on it where, like, okay, these 3 or 4 antigens are working. This antigen is causing a regulatory response. Let's take it out, synthesize a new one, and monitor. So we're playing tennis with the cancer as opposed to just trying to hit an ace. And I think also if we get this environment set up, with these pipelines, these tools in one place, it makes it. We want to create an environment where, an army of people can get in, get up to speed, and leverage the all the data and tools we have available to us to do science. The reason why we got into these fields to begin with was to push the ball forward.